Okay, so let's look at some important biological macromolecules. And some of these are going to be important because they're the main food sources that we ingest. Okay, and another one is going to be the main component of genetic information. Okay, so let's look at carbohydrates first. So carbohydrates that we typically will find in many food sources, just look at the labels on any food sources that you eat. You're almost invariably going to see carbohydrates in there. And carbohydrates are just long strands of sugar molecules that are bound together. So in actual fact, when you're eating a bowl of pasta, you could almost see it as eating a bowl of sugar. Okay? Because the body is going to break down the carbohydrate strands and it's going to break it down into simple sugars, which it's going to use as energy. Okay? And if it doesn't use it as energy, it's going to take the glucose molecules and it's going to store it for later energy. And where does the body store sugars? It makes its own strands of long sugar molecules called glycogen. And glycogen is stored in the liver. Okay? What about lipids, which are typically triglycerides? So triglycerides we'll see a molecule of coming up. But so there's three fatty acid chains that are attached together with a glycerol molecule. And that's what we would call a triglyceride, which is a fat molecule. Okay? Another important component of our food, and the body can definitely break this down as well, or it can store it, right? It can store it in fat cells around the body. If it doesn't want to store it, it can break it down. And what's it going to break it down into? It's going to break it down into fatty acids, which are the single chains that are in the lipid. And then finally, it'll break it down even further so that it can be used for energy. Okay? What about proteins that you eat? So proteins are usually found in vegetables, lentils, all kinds of food sources, muscle, so meat is high in protein. And the protein, again, that you eat will be broken down to its individual components, which are called amino acids. And the body can take those amino acids that you've eaten in the form of protein and make its own proteins that it wants to make. Okay, so all of these can be eaten, all of them can be broken down and used for energy, and all of them can be broken down and used to be stored. So carbohydrates stored as glycogen, lipids stored as fat in fat cells, proteins are broken down to individual amino acids, and then they're used to rebuild or to build new proteins in the body. Okay, and most of this is taking place in a very large organ of the body that's basically the metabolic center of the body called the liver. Okay. Nucleotides. Well, nucleotides are the bases, so the small building blocks of DNA and RNA. And DNA, so deoxyribose nucleic acid, and RNA are the genetic information of the body, and we'll talk more about that. Okay. Okay, so we'll just continue along the same lines here because these are important concepts, and then I'll show you the molecular structure of each one so it makes a bit more sense. But as mentioned, carbohydrates are really just long strands of sugar in the body, in your food source, right? So what are carbohydrates made of? Long strands of carbon, hydrogen, and oxygen. Okay, so your meal of pasta was really just long strands of carbon, hydrogen, and oxygen that can be broken down into simple sugars that can be used as energy in the body. Okay, or if the body says, I have enough energy right now, I'm going to store my sugar, my glucose, as energy for later on. It can store it as glycogen in the liver, and glycogen is just long strands of sugars. Okay, if your sugar stores were full, so your glycogen stores were full in your liver, the body can say, you know what, I have extra sugar, I'm going to convert the sugar or the glucose to fat. I'm going to make fat, and I'm going to store fat in my fat cells, in my adipose tissue. Okay, so the body wants to keep all forms of energy in case you decide not to eat one day, and then it can utilize its stores. Okay, so the main sugar store in the body is glycogen, and it typically will last for days if you're not eating. Your body will just gradually break down glycogen into glucose and use its stores of glycogen. Okay, what about lipids? Well, lipids are just fats that are eaten. They also make up the main. They are the main components of the cell membrane. Remember, phospholipids, right? With that phosphate head and the lipid tail, they make up the cell membrane of every cell in the body. But fats that are eaten, again, they're made up of carbon, hydrogen, and oxygen, right? In chains. The body will break them down, and it will either use them as energy or store them in fat cells, okay? And use them for another day when you haven't eaten enough or your body wants to mobilize the fat. Nucleic acids, as I mentioned, again, look at the typical elements that all of these are made of. Nucleic acids are made of carbon, hydrogen, oxygen, okay, just like lipids and carbohydrates, but they also have nitrogen and phosphorus, and they are the building blocks of every cell's DNA and RNA, which is the genetic information of the individual cell. Okay, so basically every cell in your body has DNA in the nucleus, and the DNA is the genetic information that sends out RNA as a messenger to make specific proteins, and we'll talk about that more. Okay. How about proteins? Well, proteins are just long strands of amino acids, just like carbohydrates were long strands of sugars. And amino acids, they're all made up of carbon, hydrogen, oxygen, and they have a nitrogen base with them. So when you eat proteins, the body can say, you know what, I want to break down that protein right down to the amino acids that it was made of, and I want to use those amino acids to make my own proteins that I need in the body. So your liver might say, I need to make more albumin, which is a very large protein in the body, and it'll break down the protein that you just ate, use the amino acids to make albumin, or an assortment of many other proteins in the body. Remember, we spoke about enzymes, which are proteins. There's so many proteins in the body that are necessary for all the different functions that the body has. Okay, so you'll have muscle proteins. So whether it's skeletal muscle, cardiac muscle, you need proteins for them to function. So you're going to come across proteins all the time when we're talking about either what people are eating or proteins that are made by the body for specific functions. Okay, so let's just look at some molecules and how they lead to other molecules. So amino acids, those are the building blocks of proteins. Okay, there's 21 amino acids that the body typically uses to make all of its proteins. 9 of the amino acids are what are called essential amino acids, meaning the body can't make them and you need to get them in your diet. And with those 21 amino acids, there's basically an infinite assortment of proteins that can be made, because the amino acid sequences will be different and the lengths of the proteins will be different. So for example, insulin, which is a hormone secreted by the pancreas, is a tiny protein. It has a very small number of amino acids. Albumin, which is a large protein, has a long sequence of amino acids. Okay, so with those 21 amino acids, you can make a large assortment of proteins. What about fatty acids? Well, fatty acids you can get in your diet, right? The molecule above is actually, there's brackets there because it should be showing a long strand of a carbon backbone. So there might be, say, 11 carbons going out, or 12 carbons. So it has the fatty acid will be a main component of the cell membrane in the form of phospholipids. So how do you get a phospholipid? Well, with the fatty acid combining with a glycerol group and a phosphate to become phospholipids. And the phosphate is the hydrophilic part of it that's facing the water, which is on the outside of the cell and on the inside of the cell. And that's why you have a biphospholipid layer, so that the fatty acid hydrophobic tails are within the cell membrane, hiding from the water. Okay, carbohydrates, right? So carbohydrates are just long strands of sugar, and they're broken down into monosaccharides. So a monosaccharide, for example, is glucose or fructose. It was originally in a long strand of carbohydrate, and enzymes broke it down to monosaccharides. And then the body can store it again, say as glycogen, as a polysaccharide, okay, for use as energy later on in case it needs it. Okay, so you always have glycogen stores in your body. A nucleotide base, well, the bases are that molecule above. You don't need to memorize the molecule, just understand the concept. So a nucleotide base is going to be the building block of DNA and RNA. And we'll get into that in more detail. So it makes more sense. Okay? So you have amino acids to make proteins, fatty acids to store fat, and also to make the phospholipid membrane, carbohydrates that'll break down to monosaccharides such as glucose, be used for the body as energy, and also to be stored as glycogen, and nucleotide bases to be used to make DNA and RNA. Okay, so this slide is just giving you an example of a protein, just to solidify your understanding. And a protein is long strands of amino acids, right? And of the 21 amino acids, they'll just combine in a sequence to make a certain protein. Some are longer, some are shorter, okay? And some act as hormones, some act as carrier proteins in the bloodstream, some act as muscle proteins. There's just an endless amount of different proteins that the body will use as basically building blocks of the body. Okay, so your bones will actually, the matrix of your bones, which is calcified with calcium phosphate, will have proteins within it that's also giving it strength. Okay, your ligaments, your collagen, which is a protein in the soft tissue all over your body, is giving your tissue strength because it's a protein. Okay, so when a protein is made, it doesn't exist like a long strand of hair because of the different electron forces, positive and negative. It's going to cause the protein to fold on itself and to turn into very specific structures. So as mentioned before, protein can be eaten, whether it's in the form of meat, beans, dairy. The body will take that, usually just break them down back to the amino acids, because the body wants its own proteins. And once it's made its own long strand of proteins, the proteins themselves, because of different electron charges, will fold and turn and have a very specific shape to them. Okay, so where do these proteins come from? Like how are they made in the body? Well, that's where the DNA comes in, right? So the DNA is in your nucleus. The DNA sends a message out to the cytoplasm of the cell in the form of messenger RNA or mRNA. And the mRNA is going to be the messenger that has a sequence of nucleotides on it. And when that attaches to ribosomes, which we'll talk about coming up, the ribosome can go along the mRNA and start to make a protein or a peptide. And the mRNA is basically telling the ribosome which amino acid sequence it wants exactly, because it's the information that has come from the nucleus, the DNA. Okay, so the sequence is DNA. The DNA makes a copy of mRNA. The mRNA goes into the cytoplasm, and it dictates which specific protein it would like to be made, which sequence of amino acids, and exactly how long the protein is going to be. So if we were just looking at the sequence of amino acids of a protein, we would just be talking about the primary structure, right? Just tell me the amino acids of a protein, okay? But that tells you nothing about the structure of the protein, because there's 21 amino acids, and they can combine in so many different sequences, and each one of them can have slightly different charges. The protein starts to fold on itself. And as it folds on itself, say in sheets or in helixes, it twists and turns and becomes a very specific shape. And then finally, the sheets or the helixes can combine together in a three-dimensional protein. Okay, and when one protein combines with another protein, they attach to each other, they become a quaternary protein structure. So I just think of it as, okay, I have a strand of hair, which is the protein amino acid sequence. Okay, as it starts to turn on itself and twist, because of some electric charges, it's becoming a secondary protein. And then as it really turns and folds on itself, it's forming a tertiary protein structure, which is really important, because remember, when we're talking about proteins, they need a specific structure. Say, as an enzyme, they need a specific structure to allow the substrate to fit right onto them. Okay? So proteins are often in a tertiary structure in the body. They're not in these long strands just hanging out. Okay? And finally, when one protein interacts with another protein in the body, they can form very complex quaternary structures. That's just the concept here. Okay? Okay, so what can damage a protein? Which is kind of important, right? Because we don't really want to damage proteins in our body. But if we're talking about the body, what can harm proteins? Well, heat, radiation, alcohol, or acids. So when we were talking about acidosis in the body, well, that's going to harm the protein shape. It's going to alter protein shapes in the bloodstream and in cells. Well, that's pretty serious. That's going to harm cells. It's going to harm the metabolic processes of the body. And that's why acidosis can be so deadly. Radiation, how about sunburns? Well, sunburns damage cells. They damage the proteins of the cells. They kill cells. That's why the skin sloughs off, and they actually alter and damage the DNA in the cell as well. Okay. What about heat? What if you have a very, very high fever? That can alter the functioning of proteins in your body as well. Okay. So if we were just to talk about proteins, say in a laboratory, how could we denature the protein? Meaning, we can alter its shape right back just to a single strand of its amino acids. Well, we can heat it up, we can radiate it, we can put it into some alcohol, or we can put it into some kind of acid. And in this scenario, we're looking at an egg that's basically protein, a lot of albumin, and the protein's being heated up and it's being denatured, and that's why it's turning white, right? And that's an irreversible process. Okay, in the body, high temperatures, high fevers, acidosis can cause enzymes and other proteins to be less functional. Okay, so this is just, the egg is just really an example of a protein that's denaturing from heat. So what are some protein functions in the body? Well, the first is that we eat protein as an energy source, right? So from the food that we ingest. And as I mentioned before, typically the body breaks down the proteins in the gastrointestinal tract, and it'll break it down into little peptides. So peptides are just small proteins, and it'll absorb the small peptides, or if it's broken down even more, it'll absorb the amino acids and take them and deliver them to the liver. And the liver will typically use those amino acids to build its own proteins in the body. Okay? Enzymes, well, those are just a type of protein that the body has made to be used as a catalyst in metabolic reactions in the body. Okay, an example here is a protein in the stomach called pepsin. Well, what's pepsin? It's a digestive protein, right? So it's a digestive enzyme. It'll accelerate the breakdown of certain proteins in the gastrointestinal tract. So as long as you remember that enzymes are all over the body to assist and accelerate metabolic processes, okay, which is really important for the body to keep up with all the metabolism that's taking place in the body. Okay, proteins can be hormones. What are hormones? Well, they're messages or they're messengers from one part of the body to another. So insulin that's secreted by the pancreas is used to communicate with cells to allow glucose to get into cells and be used as energy. Okay, insulin is also a signal to the liver to tell the liver to start making glycogen because there's lots of glucose in the bloodstream. Okay, so there's all kinds of hormones in the body that we'll talk about. Structural proteins that give your body structure, such as collagen that we mentioned. So if you were to pinch your skin and bring up some tissue, you're definitely lifting up some elastin and some collagen between your fingers. So those are structural proteins. What about protective proteins? So your immune system makes all kinds of proteins that are used to communicate with other parts of the immune system. And it also produces antibodies, which are basically like snipers that go and attach to antigens or foreign substances to destroy them. So the antibodies are released by certain cells in your immune system. They go and attach to foreign substances and tag them and destroy them. Okay, so those are protective proteins in the immune system. Okay, how about storage proteins? So the body doesn't just like accepting things into the body, like iron or all kinds of. It wants to maintain control in the body, and it'll use proteins to chauffeur different chemicals in the bloodstream, for example, or to store certain elements or molecules in the body. And one of them is iron. So iron gets stored in the liver with a protein called ferritin. Okay, so proteins can be used for storage. What about contractile proteins? Actin and myosin in your muscles. Okay, they interact with each other to allow contraction of muscles. Transport proteins, albumin. albumin's this huge molecule that floats around in your bloodstream. It transports a lot of molecules attached to albumin. Certain drugs are protein bound. Actually, most drugs that patients take are protein bound. Again, because the body doesn't like just chemicals floating by themselves. So there's many transport proteins. Clotting proteins. So basically all of your clotting factors, so we call them clotting factors, that end up helping your blood clot, are proteins that are made in your liver. Okay? Almost any protein you can think of has been produced in your liver. The liver is the major factory of the body. Okay? What about proteins that are in every cell membrane in your body, that are used for transporting molecules across the cell membrane, or that are receptors on the cell membrane waiting for signals to attach to them to tell the cell what to do, right? So that phospholipid membrane is filled with all kinds of proteins. Some of them are channels to allow different molecules through to inside the cell. Some of them are receptors waiting for signals, say from hormones. So proteins have such a huge and varied function in the body. Okay, so let's say we're looking at carbohydrates now. Well, let's look at someone eating a long strand of starch in the form of a hamburger. Well, a hamburger is going to have lots of protein, but the bun is going to have a lot of carbohydrate. So when you eat the carbohydrate, which is in the form of starch, which is just a carbohydrate, gets into the gastrointestinal system. The gastrointestinal tract is going to break it down into small sugar molecules, different types. So a disaccharide is just two sugar molecules attached. A monosaccharide is a single sugar molecule. And glucose is an example of a monosaccharide. So in your gastrointestinal tract, the starch will be broken down into smaller and smaller components, such as disaccharides and monosaccharides, that will get absorbed and then can be stored as glycogen in the liver or in the muscle cells if it's not required for energy immediately. Okay, so here's just some examples of simple sugars or simple carbohydrates, meaning they're very short in length. There's usually just one or two sugar molecules attached, and complex carbohydrates, which we would use, for example, starch as an example. And a lot of foods that we eat are complex carbohydrates. A simple carbohydrate that we would ingest would be, say, a sugar drink, such as Coca-Cola, right? That'll just have simple carbohydrates in them. So the body will take the Coke with the sugar in it. It'll easily break it down in maybe one enzyme process. If it's just straight fructose or glucose, it doesn't even have to do that. It just absorbs the fructose and glucose, and the body decides, I have way too much fructose and glucose. I'm going to store the glucose as glycogen. Or if I have too much glycogen, I'm just going to take the glucose and convert it to fat and store it in my fat cells. Okay? So simple sugars are typically just one or two sugar molecules. And if it's two sugar molecules, they're attached with a bond, right? So sucrose, lactose, maltose, those are all just simple sugars you can eat. Complex carbohydrates are longer strands of simple sugars. So starches, pasta as an example, right? They're just longer strands. Now there's some carbohydrates that we can't break down because we don't have the enzymes to break them down. So for example, animals such as cows or horses can derive sugar from eating grass. We don't have the enzymes to break down grass. So we don't have the enzymes to break down certain fibers, so we can't use them as energy. But there's many enzymes where we do have that can break down complex carbohydrates. And a perfect example of that is wheat, bread, pastas. We can break them down to simple carbohydrates and use them as energy, or store them as glycogen. Okay, so let's look at some carbohydrate details. So carbohydrates are typically spoken about in the setting of ingestion, so for energy. And let's just talk about how they're digested a bit, right? So when we eat carbohydrates, whether it's in the form of complex carbohydrates or simple sugars, which really don't need a lot of breaking down, except in the small intestine into even smaller molecules such as monosaccharides. But let's just say we have some starch or carbohydrate like bread or pasta. The digestive process starts in the mouth, right? Because saliva has amylase, which is an enzyme that breaks down carbohydrates. At least it starts to break down carbohydrates, so that by the time the carbohydrates arrive in the stomach, they're already partially digested into polysaccharides, which are still chains of sugars, but not as large, and then much smaller carbohydrates and sugars such as sucrose, lactose, and maltose. And those are just sugars with two sugars attached, right? So sucrose, lactose, and maltose can easily be broken down in the small intestine. So anyways, once they arrive into the stomach, there's very little digestion that occurs in the stomach for carbohydrates. It's a very acidic environment. The stomach has multiple layers of muscle that can churn the food and also move it forward into the small intestine, where there's pancreatic amylases. So the pancreas can secrete digestive enzymes to break down carbohydrates, to break down fats, which would be called lipases, and to break down proteins, which will be called proteases. So pancreatic amylase will continue the breakdown process until you essentially have monosaccharides, just single sugars, such as galactose, fructose, and glucose. And they easily will get absorbed into the bloodstream and travel to the liver, right? So most blood from the intestines, whether it's the stomach, the small intestine, or the large intestine, travels back to the liver through what's called the portal system or the portal vein. So all of these areas, the blood supply, go back to the liver for processing. The liver doesn't want to let in anything that hasn't been processed a bit. It wants to check out all the molecules that are in the digestive system. Okay, so the portal vein delivers, whether it's carbohydrates, proteins, fats, medications, almost anything will go to the liver first for processing. Okay, now the sugars that arrive into the bloodstream or into the liver can then be stored in glycogen as glycogen, whether it's in muscle cells or in the liver. And then when needed, the glycogen can be broken down and used to produce or turned into glucose first and used to produce the energy currency of the body called adenosine triphosphate, which is ATP. So glucose goes through a breakdown process. The first part of the breakdown process is called glycolysis. There'll be much more detail on that in coming modules. And then it goes through a cycle in the mitochondria called the Krebs cycle, where a lot of ATP, a lot of energy can be obtained by the breakdown of glucose. Okay, so let's talk about lipids a bit. So lipids or fats can be eaten and digested for storage or for energy use. But lipids also have multiple roles in the body as well, right? So the body will make lipids to perform certain roles that we'll discuss in the following slide. So when we see the term lipid, we usually think of, oh, okay, so lipids are often in the cell membrane. Yeah, phospholipids make up our cell membrane. When we're describing a lipid, it just depends on how many fatty acid chains they have that are hanging off the glycerol. So monoglyceride, diglyceride, or triglyceride lipids, just dependent on the fatty acid chains. Okay, we'll go to the next slide and we'll look at some uses of lipids in the body. Okay, so what does the body use lipids for? So first of all, so fats or lipids are a source of energy that are stored in adipose cells as triglycerides, so 3 fatty acid chains. Often when you eat fats, fat-soluble vitamins come with the fat. Which are the fat-soluble vitamins? A, D, E, and K. Okay, and you'll learn much more about the vitamins as modules go on. But those are the fat-soluble vitamins. So you can imagine, for example, if you weren't able to absorb fat for some disease in the gastrointestinal tract. Well, not only are you going to not be able to absorb fat, which is energy, and likely you'll lose weight, and the fat will go into the stool. And fat tends to be oily, greasy, and it's lighter than water, so it'll float on water. So if you're not absorbing fat, you'll have greasy stools. Okay, but at the same time, you're not going to be absorbing your fat-soluble vitamins, such as vitamin A, vitamin D, vitamin E, and vitamin K. And each one of those have different roles in the body. Okay? Insulation. So fat deposits under our skin as adipose tissue provide insulation to us to regulate temperature. Okay, so that's an important role for fat. Protection. A lot of the cushioning of our body or even around organs are performed by fats. Okay, it's definitely the major component of cell membranes. which are phospholipids, right? So the phosphate head with the lipid chains going inwards. Fats provide a lot of covering and insulation for nerves, which is called the myelin sheath, right? Which allows for faster conduction of action potentials down nerves. Fats are heavily involved also in production of hormones, steroid hormones in particular, because steroid hormones are based on cholesterol. So cholesterol is the main base for which certain hormones called steroid hormones are produced. And that would include estrogen, progesterone, and testosterone. So you're going to hear about protein-based hormones, and you're going to hear about steroid-based hormones in the body and the functions that they perform. Okay, so let's look at one of the main reasons for lipids, and that is to have a cell membrane on every cell in your body, right? To separate it from the extracellular space or fluid, and have an intracellular space and fluid, which is called the cytoplasm. So it's called a phospholipid because you have your lipid, which is your glycerol and fatty acids, that's attached to a phosphate molecule. The phosphate and the glycerol are hydrophilic, so they face towards the extracellular or the intracellular fluid in a biphospholipid layer, and then you have your hydrophobic tails going inwards, away from any water molecules. Okay, and if you can imagine hundreds of thousands of these going in a circular fashion around a cell, you have your cell membrane. And within the cell membrane will be all kinds of proteins that are lodged in, channels, you'll have receptors. It really is an amazing system, even just one cell of the body, to have this cell membrane that then has all kinds of molecules floating around within it to maintain communication within the cell, and also to be able to communicate through hormones with attaching to receptors with other parts of the body. So pretty amazing. Okay, and when we're talking about fatty acids that we're eating, say, or that are in the body, but let's just say that we're eating them, so fatty acids and lipids, a saturated fatty acid has no double bonds. It's fully saturated, okay? And what's it saturated with? Hydrogen ions, or hydrogen elements of hydrogen, right? So you can see in the top there, that is a fully saturated fatty acid, okay? Unsaturated means that there's double bonds, so there's some hydrogens missing. Because as long as you have 4 bonds around a carbon molecule, it's complete in terms of its electrons. So you can either have 4 hydrogen atoms around a carbon molecule, or you can see at the end of the fatty acid there's 3 hydrogen molecules and then 1 attached to another carbon. It doesn't matter. It wants 4 bonds attached to it. In the unsaturated, it's getting its 4 bonds by a double bond below, with another carbon. So if you see the red area of the unsaturated fatty acid, you can see that there's a double bond there, and it's completing the fact that those carbons still have 4 bonds. And when a fatty acid is unsaturated, it bends it. Okay, so it's not as linear as a saturated fatty acid. It tends to kink it a bit and change its direction. Okay, so those long fatty acids and a glycerol group that attach make glycerides, right? And we saw an image above monoglycerides, diglycerides, and triglycerides. And in the body, triglycerides are the most common, right? It's a form of energy storage in fat cells. Now, other parts of the body take advantage of glycerides. So there's all kinds of chemicals that are used in the body that you'll hear about that are made of glycerides. Prostaglandins. Well, prostaglandins are heavily involved in the inflammation system of the body. Leukotrienes, another group that are part of the inflammatory system of the body that you'll hear about. Okay, so in fact, prostaglandins are responsible for generating fevers. They're responsible for activating different parts of the immune system. And you'll see that certain drugs that you'll learn in pharmacology inhibit prostaglandins, such as aspirin is one of them. And there's some drugs that inhibit leukotrienes to try and reduce inflammation. Okay, so now if we look at saturated fats versus unsaturated fats, typically saturated fats are animal fats, and they're usually not as healthy for you, okay? Compared to unsaturated fats that are typically plant-based fats, nuts, seeds, and they're often a much healthier form of fat to ingest, okay? So when we look at glycerides, there's a glycerol group, a fatty acid group, and then you could make a monoglyceride, a diglyceride, or in this example, a triglyceride. Okay. Okay, so let's talk about nucleotides now. So we just finished talking about fatty acids, carbohydrates, proteins, amino acids. Now we're going to move on to the foundation of DNA and RNA, which is really the coding information in the cell, right? So DNA, deoxyribose nucleic acid, is the genetic makeup of every cell in the body, right? So DNA is a double-stranded helix, okay? And all it is long strands of nucleic acids that bind together depending on what base pair they have. So for example, on one strand, if there's adenine, adenine always binds with thymine. If there's cytosine on one strand, it's going to bind with guanine, okay? And it just goes on and on like that for thousands and thousands of base pairs, okay? So and in RNA, we'll see that RNA is typically single-stranded. And RNA goes out of the nucleus, so the DNA remains in the nucleus. A copy of DNA, can be produced and becomes mRNA, messenger RNA, which leaves the nucleus and goes into the cytoplasm. And in the cytoplasm, ribosomes attach to the RNA, the mRNA, and produce amino acid sequences that become proteins. So mRNA is the sequence that the ribosomes copy off of to make a certain protein. That's pretty amazing. Okay, so let's go on to the next slide and I'll explain it a bit more. Okay, so we spoke about the fact that there's a phosphate group on DNA or RNA. There's a sugar group, so ribose or deoxyribose, and then there's base pairs. Okay, so ribonucleic acid is RNA, and deoxyribose nucleic acid is DNA. Okay, so you have DNA and RNA. And so the phosphate that attaches to a sugar that has one of those base pairs, either an adenine, a thymine, a cytosine, or a guanine attached to it. And it just runs in a long strand of DNA, and another strand will match it as long as A base pairs with T and C base pairs with G. Okay? Now, in RNA, interestingly, there is no thymine. Okay? So G, or guanine, still base pairs with C, cytosine, but now we have another base pair, or a base molecule called uracil, and uracil pairs with adenine. Okay, so that's the one of the main differences, right? So DNA stays in the nucleus, makes copies of itself, or at least part of itself, sends out the mRNA as a messenger into the cytoplasm to be copied, and when it's being copied, it's actually producing a protein as it's being copied. Okay, that happens in the cytoplasm. Where in the cytoplasm? On the surface of what's called the rough endoplasmic reticulum. Okay, and we'll come to that. And we'll explain that a bit more. Okay. Okay, so this is showing you an image of DNA pairing. Okay, so remember that DNA is a double helix. So there's two strands that are twisting around each other. And that's where the base pairs bind to each other, right? So guanine binds to cytosine, and adenine binds to thymine. And it continues and continues for thousands of base pairs. Now, when DNA is being copied into RNA, which is called transcription, so the process where DNA copies itself to become an mRNA is called transcription. So whenever there's a base pair on the DNA called guanine, the mRNA will have a cytosine. Whenever there's a base pair of adenine, the RNA will put a uracil on it instead of a thymine. So RNA ends up being a single strand. mRNA is a single strand of base pairs of guanine, cytosine, adenine, and uracil. No thymine, okay? And that mRNA will float out of the nucleus into the cytoplasm. It'll go to an area of the cytoplasm called the endoplasmic reticulum, where ribosomes will attach to it, and we'll see this in another image. And the ribosomes will go along the long strand of mRNA and copy the strand of mRNA into a certain protein, depending on which sequence of base pairs have been put on the mRNA. Okay, so it's pretty amazing. The mRNA is going to be copied by ribosomes to make a specific protein, and that's called translation. Okay, so transcription is the process of where DNA transcripts a copy of itself to mRNA, and translation is where mRNA is copied into a protein. Okay, so this is just showing you a short sequence on a strand of RNA. And if we're talking about messenger RNA that's been copied off of the DNA and travels with its information to the cytoplasm, in particular the endoplasmic reticulum in the cytoplasm, where it's going to be copied by ribosomes and have amino acids in a certain sequence added to each other to make a long or a potentially long protein or a short protein. Just depends on which one it's making. If it's making insulin, it's going to be a short protein. If it's making albumin, it's going to be a longer strand protein. So you can see the information is passed from from DNA to RNA to production of proteins. And there's thousands of proteins that are built in the body, all different types for all kinds of functions. So when we look at this strand here, we can see that there's a phosphate group with a sugar group, S, and then the base pair. So G, guanine, U instead of thymine, which is uracil, and then there's an A, and then there's a C. So with 4 different base pairs, the combinations, because of the different lengths of RNA and the different sequences, are infinite, right? You can make so many different sequences that basically there's an infinite way of making different proteins. Okay, so let's just look at this image, which will make a sense of the different RNAs. Well, the mRNA, the messenger RNA, has arrived from the nucleus into the cytoplasm. Okay, so that's basically, you can think of that as the strand that the ribosomes are going to attach to. And they're just going to go along, slowly along the mRNA and copy it. Ribosomes have ribosomal RNA. They don't get copied, they're just part of the ribosomes. The transfer RNA is basically coming in and with 3 base pairs determine 1 amino acid. So you can see on here that UAC on the transfer RNA, which is linking to the mRNA sequence of AUG, codes for a certain amino acid, methionine. Okay, and it just goes on like that, and on and on and on. So the next sequence, depending on what it is, will bring in a certain amino acid on a transfer RNA that will attach to the methionine. And then the next one, and then the next one, until the final entire protein has been built, which is from the copied mRNA. It really is an amazing process in the body. Okay, so next week we're going to spend more time looking into the cell structure, talking about the different organelles and the functions of the organelles of the cell. We're going to spend some time on cell membranes and get a much better feel of how all the different organelles function and cooperate with each other to to help keep the cell healthy and functioning normally. Okay, so until next week, have a good week, and I will talk to you then. Okay, bye-bye.